In this section, we'll discuss factorial experiments for the ANOVA or analysis of variance hypothesis. In factorial experiments, we have two factors and both are treatment factors, as opposed to the previous example where one factor was a treatment factor, the other was a block factor, which we were not interested in. We were interested in only the treatment factor. Here we have two treatment factors and we are interested in both of them. So we set up an experiment where we have A levels of factor A, okay, treatment level 1, 2, all the way through level A, lowercase a. Then we have factor B going from level 1, level 2 through level B. Now we have intersection between a level in factor A and a level in factor B, and that intersection is what we call is the population. So from each of these populations, we take exactly the same number of sample observations. We'll call that R. So that's called replication. So we have one sample here for factor A level 1, factor B level 1, and then another sample here, factor A level 1, factor B level 2, and so on and on and on. So if you have B levels of factor B, A levels of factor A, then we have A times B intersecting populations. And each population we take a sample of size R, then we have A times B times R as the total number of observations. Now we can compute sample mean for each cell. So you get x bar sum 1 1, x bar sum 1 2, x bar sum 1a, and so on. And you find the mean for each column. You get x bar sub dot 1, x bar sub dot 2, and so on. And mean for each row, that will be treatment level B, x bar sub dot 1, x bar sub dot 2, and then of course the overall mean is x double bar. So in this way, okay, when we set up the experiment in this way, so we have A levels of factor A, B levels of factor B, R is the number of replications for each combination, that is the sample size for each intersecting cell, and n sub t is the total number of observations in the experiment. That will be the product of these three, A times B times R. Now we can look at each of these intersecting cells as a population, each having a mean. So we have mu sub 1 1 for level 1 of factor A, level 1 of factor B. Mu sub 1 2 will be level 1 of factor B, level 2 of factor A, and so on. Now if you take the mean of the first level of B, then you get mu sub 1 row 1 dot is averaged out all the columns for row 1, and mu sub 2 dot and mu sub B dot for the last level of B. Similarly, you have for each column mean mu sub dot 1 first column, mu sub dot 2 second column, and so on till mu sub dot A. Given that we have all of these different populations and means for different rows and different columns, then the hypothesis that we want to test, since both the factors are treatment factors, we have one set of hypotheses for factor A, which goes in this fashion. You take factor A means mu dot 1, mu sub dot 2 through mu sub dot A. So the mean of all the levels of treatment factor A are same, against at least one is different. So that is one set of hypotheses. The second set of hypotheses for factor B. So all the means of factor B are the same. Mu sub 1 dot, mu sub 2 dot, and so on. Against the alternative, not all of them are equal. Now there is a third set of hypotheses, and that is called interaction between A and B. Interaction between A and B, are there differences that are different because of the combination of levels of A and B? So here the null hypothesis is there is no interaction effect between A and B, between factors A and B. And the alternative is that there is interaction between the treatment levels of A and B. Now, to test these three hypotheses, we have the same three assumptions. Each population, that is for each intersection of level A and level B, the response variable is normally distributed and all the variances are same and the sample observations are independent. And under these assumptions, we will take samples and we will run Excel data analysis command ANOVA 2-factor with replications. That will give us the results with which we can test the three hypotheses. And when we run the data analysis command, we will generate sum of squares. The total sum of squares is the sum of sum of squares due to factor A plus sum of squares due to factor B plus sum of squares due to the interaction between A and B, and then the residual or error sum of squares. So SST is the total sum of squares, SSA is sum of squares due to factor A, SSB is sum of squares due to factor B, and then due to intersection, sum of squares due to error. These sums of squares can now be summarized into an ANOVA table. So we have factor A in the first row, sum of squares for factor A is SSA. The degrees of freedom is A minus 1, where A is the number of levels of factor A. So divide that into SSA, we get mean square due to factor A MSA. Do the same thing for factor B. Now number of levels of factor B is lowercase b, b minus 1 is its degrees of freedom divided into SSB, you get MSB. And then sum of squares for interaction AB, the degrees of freedom is the product of these two degrees of freedoms, A minus 1 times B minus 1, and divide that into SSAB, you get the mean square of interaction AB. And SSE, the degrees of freedom is A times B times R minus 1, R being the number of replications. Divide that into SSE, you get MSE. And note that the sum of the degrees of freedoms of these four should be equal to n sub t minus 1, which is the degrees of freedom for SST. Now we have three hypotheses, factor A is significant or not, factor B is significant or not, and then interaction is significant or not. So for each of these three tests, you take the MSE divided into the corresponding mean square. So MSA over MSE will give you the F statistic for the first hypothesis that factor A is significant or not. So MSB divided by MSE is the F statistic for factor B, and MSAB divided by MSE is the F statistic for interaction. Now we use the f dot dis dot RT function to find the p-value for each of these three cases, and using the p-value then we can conclude the three tests. In the next video we look at a numerical example of the factorial design.